Hello, I'm Nancy Neutrophil, and welcome to HIN Humoral Immunity News on Channel 3, live from the lymphatic system. Today, our third line of defense is preparing a war against the attack of the Toxoplasma gondii cell. Here with us, we have Dr. Gooby to explain the tactics of the humoral immunity response.
Thanks, Nance. The humoral immunity response works alongside the cell-mediated response in an attempt to combat against any intruding antigens. Specific B cells are triggered to copy and spew out antibodies to match the antigen attacking.
Wow. Thrilling. How about you take us back to the beginning to understand what is actually going on?
Will do. So first, a cell of the immune system, usually a macrophage, engulfs an antigen through phagocytosis. It is broken down by lysosome digestive enzymes and presented at the surface of the cell by MHC2, a molecule found within an APC, or antigen-presenting cell. This is known as the activation phase.
We've got Helen T. standing by in the thymus to explain the role of helper T cells in the immune response. Helen?
Hey, y'all! Here we are in the thymus, the birthplace of all T cells. When an antigen gets past the first and second lines of the defense, helper T cells with its receptors are called upon to bind with the presented antigen on the surface of the cell. These helper T cells are specific in interacting with B cells and the fragments of the antigen. Once the T cell receptors and the MHC antigen complexes join in the lock and key relationship, the macrophage is triggered to release interleukin-1. This activates the T cell to proliferate and clone, producing more of that specific T cell. Now I'm going to send it back to you guys in the studio in the next step, which makes this response completely unique from cell-mediated.
While this is happening, B cells are simultaneously recognizing and engulfing the same antigen by IgM receptors, which are specific to this foreign body. The antigen and IgM receptor complex is brought into the cell by receptor-mediated endocytosis. The antigen is digested by the lysosome enzymes found within the cell. Once these two cells join, the T cell is triggered to release cytokines, which instruct the B cell to multiply. These B cells either clone into plasma cells, otherwise known as effector cells, or memory cells.
We've got Betty B. standing by along the blood vessels for more on this subject.
Thanks, guys! As you know, I'm Betsy B cell here in the blood vessel. With these cells, the memory B cells can eventually transform into plasma cells. But for now, let's keep it simple and discuss them separately. Memory cells, which are important in the secondary immune response, hasten the body's reaction of fighting off a pathogen. On the other hand, plasma B cells, also known as effector cells, release antibodies. This process of differentiation of B cells is known as clonal selection. It's back to you guys for more in-depth discussion on antibodies.
Thanks, Bets. So just when you thought it couldn't get any more complicated, it does.
Oh yes it does, Nance. The antibodies produced from the plasma cells circulate through the blood, plasma, and lymph, which used to be known as humors, hence the name humoral immunity. An antibody reacts specifically with the antigen that triggered its formation and its function to inactivate the antigen. After the production of antibodies, they bind to the specific antigen which the body is fighting. Once bound, it is called an antibody-antigen complex, and the antigen must be disposed. There are 4 antibody disposal mechanisms, and the most important is called complement fixation. This is when antibodies call into the complement system for help. The complement system consists of 20 proteins that form a cascade of reactions. For example, an IgM, a specific immunoglobulin, binds to the pathogen and links 2 antibody molecules to one another. These complement proteins create a hole in the cell, called an MAC, causing ions in water to rush into the cell, causing the cell to lyse.
Wow, that's what's up, Doc. For those of you out there still a little confused, we've got it covered. Stay tuned for our upcoming recap on the humoral immunity response.